Well, thank you so much to Carla for the invitation to be here, and I also really want to thank Dr. Levine, who this award is named after. He couldn't be here today, but I met him in Connecticut, and I am really honored that he chose me as actually the first recipient of this award. I'm not that far into my lab as a early stage PI, just starting year 4, so this really helps. Every bit helps to try to get the ball rolling, and I really appreciate it. Carla mentioned to talk both about the science and about my career trajectory, so I'm going to skip through some of the slides I prepared on the science. I'll just give you the big picture of what our lab's working on, and then get to the career trajectory as well, because I think that's pretty related to a lot of the themes that have come up today. But first I'll talk about our science. And you may see some themes repeating over and over today about trying to learn from nature and imitate nature, which is kind of what our lab is trying to do as well. So our lab studies respiratory viruses, something we're all familiar with and will probably all experience at least once or twice this year. So these viruses cause 500 million illnesses a year in the US alone. Many of these are colds, common colds and sore throats and things like that. But about 2 million of these lead to serious illness with hospitalization. And also these are a major culprit in the disease asthma. Something that's come to light in the past decade or so is almost all asthma attacks in kids are triggered by these viruses, and many asthma attacks in adults. So these are viruses that affect millions of people every year, and there's a tremendous room to move on, even if you could reduce this by 10%, that's 50 million illnesses you could prevent. So we have some therapeutics, and we have the flu vaccine and a few antiviral drugs against flu, but basically we do not have much in our armamentarium against these viruses. So our lab is trying to figure out how can we solve this problem by coming at it in a new direction. And so what's the challenge? Well, you can find if you work on the common cold, you can find many cartoons about not having a cure for it, and this is just one of them. But why don't we have a cure for it? Well, the reason is because the common cold and the other infections caused by respiratory viruses seem to us like it's one illness because the symptoms are pretty similar no matter what virus is causing it. But actually, there's a lot of different viruses that cause the same symptoms. There's at least, depending on how you count, there's 10 or 20 common respiratory viruses like the flu that cause similar symptoms. The most common of these is rhinovirus, which is represented by the purple on the pie chart here. And then if you look at rhinovirus in more detail, you have to blow that out because there's more than 100 different flavors of rhinovirus called serotypes that look different to the human immune system. So even within one family of viruses, there's many different viruses that we're actually needing to fight if we want to prevent this illness. And that's why there isn't one big pill for all of these. It's because to us, we experience the illness as being the same, but the way that we design vaccines or medications are very pathogen specific. It's going to be a vaccine that blocks this virus, or it's going to be a drug that prevents this virus from replicating, and it's not going to cover the gamut. So I got interested. I had a PhD in basic immunology and medical training and diagnostic testing. And during my medical training, there was a technological revolution in the way we detect viruses, and that's how I got into this field. So when viruses were first discovered in the 1950s and 60s, up until about a decade ago, our ways of trying to find them in samples like nasal swabs from humans or human tissue were very insensitive and maybe could detect a virus 5% of the time the virus was there. Now we have PCR and genetic-based methods, which, a long story short, are extremely sensitive and specific methods for picking up viruses in clinical samples, which are basically considered 100% sensitive and specific, and now we can easily see viruses. So this came into being when I was a resident, and we're supposed to be ones interpreting these tests, and all of a sudden, not only are we seeing that we can detect viruses in people who are sick, but we're seeing that we can detect viruses in people who are healthy. And not just a few people who are healthy. So this is a study that was done in Utah in 2015 where they actually convinced families with young children to get nasal swabs on all family members every week for a year, which is pretty impressive. But some really interesting data came out of this. And if you look at the top line here, the black symbols represent number of virus-positive weeks per year on average. So the top line is kids under 5. These kids had a virus in their airway 26 weeks a year, so half the time. Okay, now we know kids get sick a lot, but they're not sick half the year. And if you dial down on the data, the bottom line is there's many, many asymptomatic infections. And many of these viruses are asymptomatic 40 or 50% of the time. So one way that you can, what that makes you realize, well, first of all, when you're a resident and your job is to interpret these tests and someone calls you up to interpret that, you don't know how to interpret that, right? What does that mean? And as a scientist, it raised a very interesting question to me, like, and the question is really this: what is our body doing right so much of the time? Because this new technology for detecting viruses makes you realize we're actually getting exposed to these all the time, and actually we're really good at curing the common cold, right? Our bodies are naturally really good at doing this. We just don't know how it works, right? But we know risk factors, we know certain people who are more likely to get very sick, but we don't know the molecular mechanisms. And that's basically, long story short, what my lab is trying to figure out. So this is a big picture view of what we're trying to do. The idea is that we're all walking around, we're all exposed to respiratory viruses all the time as a factor that's just part of our environment. Some people have inappropriate response to this and they get sick. And some people get a little bit sick and get a cold, and some people get very sick and get an asthma attack. Other people have very effective antiviral defenses, and even though that virus is there, they would never know it unless they were part of that study in Utah or a study like that. So what we want to do is fill this knowledge gap. What are the molecular mechanisms that differentiate, what are the molecular mechanisms responsible for fighting these viruses that tip the balance one way or another, which infection is going to cause illness and which isn't? And so that's in a nutshell what my lab is trying to accomplish. We've had a lot of exciting results by studying the epithelium, or the lining of the airway, which is where these defenses happen. We've learned a lot about environmental factors that tip the balance between how effective those defenses are. And there have been a lot of interesting spin-off projects. One thing that I resonated with Dr. Levine on is that he's an inventor and has invented all kinds of diagnostics. And one thing that we realized is that the responses of those cells that form the airway lining are actually a very good diagnostic of what type of infection you have, because they've already made the diagnosis and started fighting the infection. And we have other interesting spin-offs looking at how infection with one virus may affect your susceptibility to another and things like that. So those are the types of questions that my lab is trying to answer. But I'm going to skip through some of the more detailed slides. You're welcome to visit. If you visit our lab webpage, you can learn all about this. But I want to skip through that and talk about my career trajectory, which is something I usually am very loath to talk about, actually, but I feel like it's very appropriate for this meeting. And I have an unusual career trajectory, as depicted on this slide. So right out of undergraduate, I went out to Stanford, and I got an MD and a PhD degree in the normal amount of time for those 2 degrees, which is 8 years. In reflecting back, I realized, I was the only female in that program in my year, which I didn't really process that till I put this all together for this talk. So then I was pregnant and had a child at the end of medical school, and I decided to defer my residency for a year. Then I went to do my residency training at Harvard, where all this stuff was happening with viruses that I just described and how I became interested in it. And again, so this was a program, all MD PhDs, all of us want to do academic medicine and do biomedical research. But in reflecting back, I realized I was the only female in that program the whole time that I was in it. And actually, even talking with some of the other colleagues here at lunch, I realized, I didn't tell anyone I had a child at home. I didn't tell anyone I was pregnant for most of the time. And a lot of my attending physicians did not know I had children. Well, when I was pregnant, they didn't know I already had a child at home, even at graduation. So, and then for various personal reasons, which I won't go into, part of it health, part of it the children, and so forth, I did not seek employment at the end of my residency, but I never had that in my mind as giving up on my career. I saw it as a pause. I We read a lot of articles today about people falling off the ladder. We have equal male and female in the undergrads, but then we don't have any faculty that are female. Well, I wouldn't say I fell off the ladder. I definitely stepped off the ladder. But you always hear different explanations, and I'm just giving you one case study of what my experience was. So I had another child during that time, but never in my mind did I ever think I wouldn't be a scientist. And even when I was not working at all, I told my husband, we need to move somewhere when we leave Boston, near a major academic medical center, because I'm going to go back and I'm going to do this. And he's like, okay, we'll do that, you know, at the cost of some very good job opportunities for him, and you know, a lot of personal decisions and struggles with that. Then I finally re-entered science in 2010, did a postdoc at Yale in immunobiology, and now I'm entering year 4 of my full-on tenure track position with my own lab, startup package, the whole 9 yards. The dream job that I always wanted and I feel very, very privileged to have and have a lot of enthusiasm and energy for now. But I want to take this opportunity to talk about what I thought was very helpful in getting me, I guess, to step back on the ladder and making this career happen, and what I thought were some of the obstacles, some of which are still in existence and some of which have been solved. So what mattered? Well, one very concrete thing that mattered when I was going back and trying to look for postdocs was the NIH reentry grant program, where I could come in, say, to a postdoc mentor, I'm pretty sure I can get this reentry grant. It's meant for people like me, and you're not going to have to fund me. Like, I was competitive for any lab I wanted to go to because of that, and because of my CV, even though my CV was pretty old at that point. Other thing was mentorship and environment. I looked around, I knocked on many, many doors until I found one that I felt was a good fit. And a supportive family, of course, that helps anybody in their career. Obstacles. So a major obstacle, which is still an obstacle to this day, is lack of family-friendly policies in medical training in particular. Just in September 2019, New England Journal of Medicine has a podcast where they have commentaries. And just this fall, there was a commentary in 2019 about, you know, we really should come up with a parental leave policy for medical residents. There's no uniform parental leave policy, and it's really at the discretion of the residency director and whatever people decide is okay for call coverage. You know, and this is in 2019. And if you're, not everybody cares about having children, but if you do, that's pretty daunting to think that that's completely out of your control if you sign up for one of these programs. And so this is starting to change, but it has not actually been solved even in 2019. Another thing that, if you're an academic, you'll understand this. If you're not, you won't. But the early stage investigator designation has really been a huge difficulty and obstacle for me. And that is because the NIH tries to provide a benefit to new investigators by saying you're going to get a huge break on the score you need to get your R01 grants funded, which is what you really need to survive in a position like mine. But there's this 10-year rule. If it's more than 10 years from your last degree, you're not eligible, okay? And when I first came back to work, I was completely ineligible. Now they've changed the rules, and you can apply, and you get a year credit for the kid, and this and that. But you still have to, it's an application process, you're justifying yourself. And the bottom line is this is just a gate to people with a non-traditional career path. And when I first came back, it was extremely discouraging because there's a lot of behind closed door judgments of my choices that are really affecting my ability to compete as a new investigator. And that, talk about knocking someone off the ladder if you can't get one of those grants, you know? So I really feel strongly if there's going to be an early stage investigator benefit, it should be defined by career stage and absolutely nothing else. And if our real goal is to increase diversity and inclusion in science. Otherwise, there's no other way to do this where it isn't just a blockade. That even if you can apply and appeal and scrape and beg, it's still a blockade. So those two things still could use some work. They're much better now, but still could use some work. when I was a student in the early stages of my careers, I had very few role models, and I could theoretically know about people, but I couldn't look in front of me and see people who were solving the problems I wanted to solve, who'd actually done it, and they were happy and satisfied and had a successful, ambitious career. And I have to say that that's something that I've really loved about this meeting, which is I'm looking in front of me now, and I am seeing that this problem is being solved. And the students today can look at all the people here who have done different things, made different life choices, but have had a successful, fulfilling, ambitious career in science as different role models. And I think that that's really a wonderful thing that this society does to bring us together to sort of celebrate this and to brainstorm about how we can solve some of these problems. So with that, I just want to say thank you again to Dr. Levine for the award, and thank you to the RFS for inviting me. It's really a pleasure to be here.